Thank you. Thank you for the opportunity letting me speak tonight. Does this work so quickly? Disclosures. So I was a co-founder of Knott Biosciences and a co-founder of another company called Iron Horse Diagnostics. Of course, none of the work that I'll talk about received funding from either of these two entities. So talk to you a bit about our metabolomics work that we've been doing. Started as a collaborative study in 2005, 2006, while I was still at the University of Pittsburgh, and it involved Mass General and with Merit, and a company called Metabolon, which is in North Carolina. We started collecting blood and CSF from ALS and control subjects. With the blood, we were using both heparin and EDTA-coated tubes. We found, though, that the heparin coating actually impeded some of the GC-MS results, and so we switched just to EDTA-based tubes for plasma going forward. So just a little note if anyone's interested. So again, our platform that we were using was down at Metabolon, and so it was a mass spectrometry-based approach using both gas chromatography-based mass spec, so GC-MS, and also liquid chromatography or LC-MS-MS-based methods. So again, we're looking at very small molecules, small metabolites that are detected by these spectral peaks. Obviously then, your ability to identify whatever metabolite you're looking at is dependent upon having a library of known compounds, a library of known what are those peaks and what they actually represent. So you are somewhat limited by whatever that library is. And at the time we started, the library was small, but has been growing by leaps and bounds in the last about decade. So we started out in our sort of discovery phase mode, taking samples and doing comparisons. We published a study in 2012 looking at a couple of group comparisons. So in the first, we compared 62 ALS and 69 healthy controls, and in the second, 99 ALS and 48 healthy controls. We found, we did find that one of the most important spectral peaks that was definitely identifying who had ALS and who didn't, it was only in a subset. That was kind of interesting. And of course, Metabolon had no idea what it was. It was just a peak, but it was present in ALS patients in a subset. So a lot of time was spent in purifying that peak, sequencing that peak to figure out what it was. It turned out to be a metabolite of riluzole. So we could definitely tell if you had a metabolite of riluzole in your system, you were an ALS patient. We did a sub-study, actually, that was run at MGH to figure out, well, could we wash out that signal? And so patients were washed out of all medications, vitamins, whatever non-essential meds that they were on for a week, and you would see that riluzole peak would go away. And so we could get rid of that confound with a week washout. It didn't affect most peaks, so there are a few peaks that just a week washout in patients, and some of the peaks that would appear to be somewhat interesting go away, but most of them do not, so that's good to know. And obviously, the limitation of this study was that we were comparing ALS to healthy controls, and so we didn't have the appropriate disease controls in this study. But here's a table of the metabolites that we saw, where green in this is going down, so in that particular case, the metabolite is being at lower levels in ALS patients, and if it's red, it goes up. And what's shown here are the overlap between the two study groups that we did. So we were looking for basically commonalities that we could see across the groups. So obviously, in any types of omics technologies, often you come up with an answer, and then if you try to replicate it, it goes away. And so we were most interested in those that were similar across the two studies and exhibited the same trend, either up or down. And so if we want to follow along the same storyline that Vincent was mentioning, so a couple of them included creatine, which was going up, and creatinine, which was going down in our ALS patients. But again, definitely a number of metabolites involved in amino acid metabolism, lipid metabolism, and some xenobiotics, so alterations within the hepatic system. And so the nutshell from this particular publication and study was that, yeah, we could definitely see there's hypermetabolism ongoing in the patients. There seemed to be mitochondrial dysfunction from altered mitochondrial metabolism. Markers of oxidative stress or metabolites related to oxidative stress were increased. Changes in lipid metabolism, and again, this altered hepatic function that was present. Now, ongoing at the same time was some work done by Anna Wolkinen and colleagues looking at CSF-based metabolomics. And she published 2 papers in the last couple of years. And in the first, she was doing, again, GCMS on the CSF from about 78 ALS patients, 17 of which were familial patients with SOD1 mutations, and matching healthy controls. And essentially what she found was a signature of about 120 peaks. Many of them weren't identified. Some of them were. They're predominantly amino acid metabolites. But that signature could stratify the ALS patients away from the healthy controls and also could correlate the site of disease onset within the patients. What she also noticed, which was quite interesting, was that she could see differences between the metabolic signature between sporadic ALS patients and familial ALS patients. And she also saw that, again, glutamic acid and creatine were also decreased in this study. And then in the second study, which came out last year, she specifically wanted to further explore the potential differences between sporadic and familial forms of the disease. And so she compared 16 familial patients that harbored SOD1 mutations, and there were 6 different mutations within that group, to both sporadic patients and familial patients that harbored other mutations, non-SOD1. And what she found was that, again, there was differences between sporadic and familial, but there was even differences amongst the familial. Those with SOD1 mutations had a slightly different signature with respect to metabolites as opposed to familial patients with non-SOD1 mutations. And if you're homozygous for the D90A mutation of SOD1, that was even distinct, or further distinct, from other patients that harbor SOD1 mutations. So quite interesting. It's suggesting that underlying metabolic changes that are ongoing in familial and sporadic patients are different, and even small differences in the metabolism of people that harbor mutations within one gene, SOD1. Now, of course, I want to mention, finding this type of discovery efforts for any of the omics technologies is actually the most easy part of the long-term endeavor. So discovery is easy, but the next steps after that become more and more challenging and cumbersome. Really, the next step that one should do then is really look at whatever your assay is for whatever metabolite, protein, gene, whatever you're doing, and really work on assay development and validating whatever that assay is. You need performance standards for whatever the assay is you wish to perform going forward, and you want to verify whatever your initial findings are, typically in tens to hundreds of samples. And then once you've done that and have a really robust assay, then move on to larger validation studies in a retrospective manner. A lot of people in a lot of diseases skip from from step 1 to step 3, thinking their assay works really well, and at the end of the day, everything falls apart, often because the assay wasn't robust. Once you've figured out this, you can go and perform large retrospective studies, and typically hundreds to thousands of individuals. If that's positive, then you can go on forward into a larger prospective study where now you can actually start asking the real questions of clinical utility for whatever the biomarker might be. And again, you need hundreds to thousands of samples for that. So now I'm going to mention a bit of work that's been ongoing, continuing the relationship that we've had with Metabolon, and focusing again on plasma-based metabolomics. So in this study, again, Metabolon have been working on their platform now for a number of years. We thought it was quite robust. We perform a lot of quality of control measures throughout every study that we do. So for instance, when you're doing any of these omics or mass spec-based approaches, you're running the instrument for hours to days. And so you get some fluctuation within the instrument itself over time that you got to control for. So in this case, we're running a pooled QC sample of plasma that we have spiked in at 30 known molecules, 30 metabolites at known concentrations. And we follow that throughout the day at different runs and over time to look for any changes within the instrumentation and the running of the samples themselves. And so here we're looking at 172 ALS patients. We reduced our healthy controls down to 50, and then we added 73 appropriate disease mimics. Also, what we've incorporated is SOPs for how samples are collected, how they're processed, how they're stored, how they're shipped. All the sites that we collected samples, so a lot of the NEALS consortium of sites, all followed the same protocols. Everyone received barcoded collection kits. Many of you were involved in this endeavor. And it actually works quite well. So we can do QC on individual samples across different sites and get similar results. So we performed, again, both GC and LC-based mass spectrometry on these samples looking at metabolites. And we could identify a number of biochemicals. We found about 280 that were at least present in over 50% of all the samples, of all the subjects. And then we used a group of 3 different algorithms to mine that data set. So a machine-based learning algorithm called LASSO, random forest, and a support vector machine algorithm to see if we could both classify each subject and then ultimately to do cross-validation studies. So just looking at some of the ROC analyses from this study, looking at ALS versus disease mimics. So by using both SVM and LASSO, we get an area under the curve of about 0.76, which isn't too bad. If we used RF, the area under the curve was a little less, 0.68. If we look a little deeper, and then, so if we take these results and then look at what's the sensitivity and specificity of looking at ALS versus mimics, it actually is only about 63% accurate. So it's pretty good at picking out who has ALS, has a little problems with some of the mimics. But again, if you're just looking at ALS versus healthy, it actually works a little better. So again, the importance of using disease mimics within the experiments. We then narrowed this down to sort of a top 32 candidates from the study, and then we used these learning algorithms to make a predictive model and ask how well can this top 32 metabolites then predict who has ALS and who doesn't. So this is a list of the metabolites here. And surprisingly, or what we thought was good, was that many of them, and again, what we saw now 3 or 4 years ago was a much smaller group of the peaks that were actually identified within the library, but many of those actually replicated now in the much larger study. So again, we could see the same thing, and good for the study, it was in the same trend. If it was up in the first study, it was up in this study. If it was down in the first study, it was down in this study. We also then looked at the measure of that metabolite and tried to correlate it with the extent of disease progression. Now, this was a static one time point per subject, and so we looked at the ALSFRS of each of the individuals and then graded what's the correlation of the level of the metabolite versus the ALSFRS measurement. And so when we did that, now if you just look at, say, creatinine at the top, there's a positive correlation, which means that as your ALSFRS measurement is actually going down, the levels of creatinine are going down. And for instance, for creatine, you have a negative correlation. So as your ALSFRS measurement is going down, the levels of creatine is actually going up. And so again, you can look across various markers or metabolites from a group of pathways by this type of analysis. And again, when we used our top 32 candidates, now we had an area under the curve, approximately about the same, but when we then in a cross-validation asked how well can it actually predict ALS versus non-ALS, we saw that the specificity was pretty good. So it was 90%. So if in fact you reached the threshold for these top 2 candidates where it said you had ALS, it was probably right. It was right 90% of the time. Sensitivity was still low, still in the upper 40s to about 60. So it wasn't very sensitive to identify ALS, but if it said you had ALS, it was probably right. So again, overall in this study, we have hyperlipidemia, a number of metabolites involved in lipid metabolism, hypermetabolism, both carbohydrate-based, amino acid-based, and altered mitochondrial energy metabolism. These are the types of metabolites we keep detecting. And so for future directions, when we're looking at sort of metabolic studies in ALS, really we need much larger both verification and then ultimately validation-based studies. We want to compare between fast and slow progressors and include disease mimics and really eliminate healthy controls from these types of studies. We're not interested in saying who has ALS and who's healthy, who has ALS, might have a similar type of disease that might look like ALS. And then again, with the fast and slow progressors, that's much more challenging because we want to do longitudinal sample analysis, and many of us are doing that. The challenges are that often when you're doing these types of longitudinal studies, you end up inadvertently selecting for slow progressors. Those are the patients that can keep coming back and providing blood or CSF over and over and over again. And the fast progressors might come into the clinic, they've already progressed to the point where it's easy to see, and they come in maybe one more time, sometimes not at all. And so you only have one or two time points. And that's a challenge for any type of study because no matter what you're looking at, you always have a straight line between 2 points. You need at least the third. And so it's a challenge, but one that we have to actually try to identify patients earlier, so even if you're fast progressor, we can include you in these types of longitudinal studies. Another great set of samples to get would be those that harbor mutations in genes associated with ALS but are pre-symptomatic. Michael Benatars has been doing this type of study for quite a while. And if you're going to do a study or a metabolomics-based study, those actually might be patients you do want to compare to healthy controls. So these are patients that have yet to exhibit symptoms that they might have ALS. They do harbor a mutation in a gene associated with ALS. So it might be interesting to compare the metabolism, the metabolomics of those patients before they actually have symptoms to healthy controls. And again, any type of omics, a metabolomics-based study, you're looking at a snapshot of the whole body metabolism that's undergoing. So no matter what the marker is, ultimately you have to figure out what's the organ and cell type of origin for that particular metabolite. And then can it be ultimately combined with some of the other omics technologies to look at larger scale pathways or systems-based biology? And obviously, we should be trying to incorporate these in clinical trials, and if you can't do that, at least collect the samples. So at the end, just acknowledging the people that have really done the work, starting for myself at the University of Pittsburgh and now in Phoenix, Arizona. So the group of individuals in my lab and Dave Lacomas, the neurologist in Pittsburgh involved. At Mass General, Merritt and James Barry, who's really taken up the lead in this for the last couple of years now. At Metabolon, the group of people that have done a wonderful job and have been very helpful in performing metabolomics on ALS patients. Our NEALS consortium of sites that has been wonderful in collecting samples, the biofluids repository that we can obtain these samples from, and then funding support from NIH, the ALS Association, Metabolon, which also provided support, and our private donors. And really just always a thanks to the patients and families that provided the samples for us. Thank you.